Hello! In this lecture series, we'll explore the accessory organs of the digestive system. In the first part, we'll discuss the overview of the accessory organs and more specific histology of the salivary glands. From the anatomic perspective, the GI accessory organs are comprised of the three pairs of the major salivary glands, which include the parotid salivary glands, which are positioned just lateral to the ramus of the mandible. These are by far the largest of the salivary glands, and they're responsible for producing a lot of watery secretions and injecting them into our oral cavity. The pair of submandibular salivary glands sit just under the mandible, and these mixed glands produce both the watery and slightly mucinous secretions into the oral cavity. And lastly, we have the sublingual salivary glands. These paired salivary glands sit just under the floor of the oral cavity and produce mostly mucinous secretions. So together, these salivary glands play an important role in keeping the oral cavity wet, protected, and clean. Next is the liver. Now this liver sits under the diaphragm and is positioned within the abdominal cavity. And I actually am going to make a quick correction here. The liver should be positioned anterior to the esophagus, which travels on the posterior aspect of the liver. At any rate, liver plays an incredibly important role in our body. It has been said that the liver provides over 200 different functions for our body, but within the scope of the digestive system, the liver functions in the filtration of a lot of blood that's coming from the GI tract before that blood is directed back into the circulatory system. Furthermore, it produces bile, which is an important emulsifying agent, which facilitate the lipid digestion and absorption. Next, we have the gallbladder. Now, I don't like this image again because the gallbladder really should be kind of attached tightly to the underside of the liver like that. So please ignore this part. At any rate, the gallbladder derives from the common embryonic origin as the liver, and its main function in the digestive system is to store excess bile that's produced by the liver and injected into the GI tract when there's a need. And lastly, we have the pancreas. This important organ sits in the retroperitoneal position. It originally formed within the peritoneal cavity, but during the process of gut tube rotation in embryonic development, this organ got pushed back to the retroperitoneal position. Therefore, we call it the secondary retroperitoneal organ. Pancreas plays an incredibly important role in producing many types of digestive enzymes that are critical for breaking down the food that travels through the GI tract so that our body is able to absorb the nutrient molecules. Furthermore, the pancreas hosts some endocrine cells that produce insulin and glucagon, which regulate the blood glucose level. Now it's time to look at these accessory organs one at a time. For salivary glands, across the three pairs, there are some general histological features that are shared. So let's discuss them first. These are all large exocrine organs organized into lobules and lobes. So if we were to take a cross-section through one of these organs and look at the histology, we'll see smaller bunch of secretory units organized into the lobules, and number of which form a bigger lobe. So these type of organizations can be a little bit arbitrary, but what helps us is that each lobules and lobes are separated by small amounts of connective tissue septa. You may remember from the epithelial tissue lecture that large exocrine glands are kind of like a bunch of grapes with a main stem and smaller branches that lead into even smaller branches, which then end up in small grapes. So imagine each grape being the secretory units and number of these grape bunches then forming a lobule. And then should we have some neighboring grape bunch, there'd be another lobule, so on and so forth. And then imagine number of these lobules together forming a lobe. And then off of another branch, we may have the similar organization. So this is a diagrammatic example of a large exocrine organ and their ductal system organization. Speaking of the secretory units are either comprised of the serous acini in the case of the parotid salivary gland, which primarily produces some watery fluids but containing some amylase. This is the enzyme that can start to break down the carbohydrates. So when we chew on something like bready, starchy materials, we start to taste sweetness in our mouth, and that is thanks to the amylase. We also have some antibodies and other molecules that are also microbial. Another type of secretory units we might find would be the tubuloacinar units. The tubuloacinar units produce not only the watery fluids with amylase and IgA, but also it produces the mucin. That's where the tubulo would come in, comprised of goblet cells. The system of ducts in these larger exocrine glands, especially in the salivary glands, are fairly well organized in that each one of these secretory acini or tubuloacini would be draining initially into smallest of ducts called the intercalated ducts, which then drain into the larger ducts called the striated ducts, which then lead into the interlobular ducts and then into the interlobar ducts, and the interlobar ducts would then all collect into one of the major ducts that drain the entirety of the salivary glands. The intercalated and striated ducts are actually positioned within the lobules. So these two types of ducts are classified as the intralobular ducts as opposed to the interlobular ducts. Inter refers to in between. So macroscopic histological image such as this in a cross section, oftentimes the interlobular ducts would be seen in between the lobules, and interlobar ducts would be seen in between the lobes, and they're physically large as you can see too. And the intercalated and striated would be so small that it may be hard to discern because they're positioned within intralobular location. So we may need to zoom in a little further to discern the intercalated and striated ducts. We also have stromal cells. These are the supporting cells within the salivary glands, and these include the myoepithelial cells that are positioned on the outside of the acini, as well as some of the smaller ducts that have contractile abilities that can help move things along, if you will. We also have some unilocular adipose cells that infiltrate into the parenchyma of the salivary glands. So wherever we see a lot of these non-staining, really tiny pockets of areas within the lobules, these are most likely the unilocular adipocytes. And we also have a lot of lymphoid cells that could be found within the stroma, including the plasma cells that produce the IgA, as well as macrophages and lymphocytes. Let's look at the ductal histology in a little more detail. Starting with the two types of the intralobular ducts, we need to zoom in into a small area within the lobule to see some of these intralobular ducts. And just to kind of orient ourselves, for the most part, this field of histological image is comprised of these secretory units, which are mostly serous acini that are producing watery secretions with proteinaceous materials in it, such as IgA, amylase, and things like that. So these secretory cells are for the most part pyramidal in shape, and they have a lot of these secretory granules in their cytoplasm, and their cytoplasm tend to be fairly large, which accounts for how far the nuclei are positioned away from each other. And now look at here. We have a cross-section of a circle with a lumen in the inside. And look at the nuclei and the distance between these nuclei. These are all intercalated ductal cell nuclei, and see how crowded they appear, which means these intercalated cells are fairly small because these aren't the cells that are producing protein products. So that's one way of distinguishing the secretory units versus one of the smallest of ductal system, which is the intercalated ducts. Here's a beautiful longitudinal cut of the intercalated duct which may actually be becoming a cross-sectional cut right here. But here's the lumen, and lining this lumen is a simple cuboidal epithelium comprised of really small cuboidal cells. And what is beautiful about this image is that we can actually see the transition between the intercalated duct and the seemingly larger duct, which appears to be lined by simple columnar epithelium. So this is the other type of the intralobular duct, which is a striated duct. Here's another striated duct with the lumen, and the composition of this duct is again a simple columnar epithelium with the cells that have a little more eosinophilia in their cytoplasm. And the reason why this duct is named striated duct is because when we look at the subnuclear compartment of these cells, so these basal compartments of the columnar cells, we see these striations. And these striations within the cytoplasm of these columnar cells formed by the cytoplasmic membrane infoldings as well as the high concentration of mitochondria that are occupying this region. So these are the two types of intralobular ducts of the salivary glands. Before we move on, let me just point out some of these stromal cells that we can actually see at this level. Here's a unilocular adipocyte. Here's another. So some unilocular adipose infiltrations can be appreciated, arterioles, capillaries, and some of these flattened nuclei outside of the acini often belong to those myoepithelial cells. Now let's look at the interlobular ducts. So we'll focus our attention on these larger ducts that are physically positioned in between the lobules of the salivary glands, and their histology looks like this. First and foremost, we're seeing a lot of these connective tissue surrounding these larger interlobular ducts. And this connective tissue, of course, contains a lot of vasculature, arterioles, venules, and once again in this beautiful slide, we can actually see the transition, the continuum of these ducts and their histology. This, I might argue, is more closer to the striated duct within the intralobular ductal system, which is continuing on to become more of a larger duct, which is continuing on and draining into this larger interlobular duct. And as for the composition of this interlobular duct, I would think that this duct too is comprised of simple columnar epithelium, although some of the larger interlobular duct could be comprised of a little more substantial lining epithelium such as striated columnar epithelium. So I wouldn't be surprised to see that down the road. Now looking at the interlobar duct, which is physically positioned in between the lobes at a higher magnification. The size of the lumen is much large. It is surrounded by much more connective tissue components, and the lining epithelium of such a large duct is the stratified columnar epithelium. So keeping these general features of the salivary glands and their ductal systems, let's move on to the individual salivary gland pairs. Here's the parotid salivary gland, which is located right here, made a transverse section through an area, and this is what the parotid salivary gland would look like under the histology at macroscopic view. The secretory units of the parotid salivary glands are actually exclusively serous acini in composition, meaning the parotid salivary glands produce nothing but mostly watery fluids containing amylase and immunoglobulin A, and no mucin. Looking at the higher magnification of the serous acini, we are seeing these typical spherical secretory units comprised of the pyramidal cells with the distinctive kind of dual staining pattern of these cells, with the basal compartments staining more basophilic due to a lot of RER indicating translation, and the apical compartment containing many secretory granules that stain eosinophilic, and these secretory granules would contain the amylase. and other microbial proteins. While we are here, let me point out one myoepithelial cell right here. It's just sitting outside of the basement membrane of the serous acinus. And now that I've pointed one out, you might be seeing some other myoepithelial cells such as these by their flattened nuclear feature. And I think this might be an intercalated duct that's cut in an longitudinal/oblique way with potentially the lumen running in the middle right here. So comprised of simple cuboidal epithelium. At any rate, when we look at a large exocrine gland with intercalated ducts and potentially even striated ducts in the nearby region, and if all the secretory units in that organ is all serous acini, then we can comfortably identify that organ as the parotid salivary gland. Next, onto the submandibular salivary gland, which is located right here under the mandible. Again, paired structures. At macroscopic cross-section, we can once again appreciate a lot of this lobular organization of the secretory units and ducts, as well as perhaps lobar organization of the secretory units and the ducts. And even though we are super zoomed out, we may be able to identify some potential ducts, the interlobar ducts probably here, interlobular ducts here, and potentially some interlobar ducts around these areas. And we can't really identify intralobular ducts from this magnification. At any rate, what sets the submandibular salivary glands apart histologically from the other types of salivary glands is in the composition of the secretory units. At higher magnification of the boxed area, we see a lot of these serous acini. So majority of the secretory units appear to be the serous acini, but we do see some minor pale staining tubule looking secretory units, which make up some minor components of the secretory units within the gland. Furthermore, we also see these characteristic demilunes, which is characterized by the mucin-secreting tubules that are capped at the end by the serous units. So the secretory units of the salivary glands are mixed. However, the acinar components by far outnumber the tubulo-acinar components, which means the secretory products of the submandibular glands are mostly watery fluids with amylase and antibodies, but in addition to that, they also produce some mucin. And before we move on, we can once again identify some adipocytes and some intercalated ducts that are transitioning into these larger ducts within the lobule. So this would be the striated duct, and this actually might be a conversion point between the intercalated to striated duct, but this is more consistent definitely with the intercalated duct. So all the commonalities are there. It's the secretory unit composition that'll help us determine that we're looking at the submandibular salivary glands. And lastly, the sublingual salivary glands. These are the paired glands that sit just under the floor of the oral cavity. And again, all the infrastructures are there, the ductal, the lobular, lobar organization. But what sets the sublingual salivary glands apart from the other types of salivary glands would be the secretory unit composition. So at higher magnification of the boxed area, what we see are the mostly mucus secreting tubular secretory units comprised of goblet cells that are quite branchy, but intermixed within this mostly mucin secreting units are occasional serous acini as well as the demilunes, again comprised of the serous secreting cells. So this type of histology indicates that again, sublingual salivary glands are mixed type of glands in that they have tubuloacini and acini, but the tubuloacini far outnumber the acinar composition, which then indicates that the secretory products of the sublingual salivary glands would be mostly mucin, but it would also have some watery fluids with amylase and antibodies. In this particular image, we're able to identify one striated duct, and there doesn't appear to be any apparent intercalated duct within this section at this time. In this macroscopic image, there's a couple of other anatomically interesting features, so let me highlight those. Here in the boxed area is a structure with a large lumen in the middle. At higher magnification, what we realize that this is also a duct. This is one of those larger ducts that drain the entirety of the sublingual glands. So look at the lumen and the lining epithelium. The lining epithelium is a beautiful example of stratified columnar epithelium, which is reinforced by the lamina propria, the connective tissue, and also some strips of smooth muscles. And another interesting anatomical feature is this boxed area. And when we look at that boxed area, higher magnification, we can appreciate that this is a lining epithelium of the oral mucosa that is comprised of the non-keratinized stratified squamous epithelium with the underlying loose connective tissue. So we can actually see the anatomy of the sublingual salivary glands from this histological tissue. In this video, we reviewed the anatomical overview of the accessory organs of the GI tract, as well as the specific histology of the salivary glands. In the second part, we'll look at the specific histology of the liver, gallbladder, and the pancreas. See you soon!